time to talk about immunology of HIV. Now, now we need to understand. I would like to start with this. The reason why I will start with this because it will make sense for you. Okay? So basically, the HIV, as you can see in this picture, um, let's start from here. Okay? So you know that our body will be exposed, via, let's say, the mucosal membrane of let's say, of the vagina, because this is how HIV is transmitted. There will be some dendritic cell in here. This dendritic cell, or the antigen presenting cell, will take the virus and basically attach or internalize or do whatever it wants. If it is attached, okay, it will present it in the lymph node. If it is internalized, it might affect the dendritic cell, and the dendritic cell will serve as reservoir for this, virus, and it will be infected for the whole life. So it's better, like if it is attached, it will be better than infecting it. Now let's say it has been attaching to the dendritic cell, it will go to the lymph node, regional lymph node. Now, some books say that the first thing it will be affected is um, is the lymphoid organ, gut lymphoid organ, and because there is a lot of memory T cells there, and memory T cell will express the CCR5, which is basically the one which is infected, which is the M tropism. Okay, M tropism, which is an early infection. We discussed this on the first lecture. So anyway, so it will be affecting this. Then it will go to the regional dendritic cell will go to the regional lymph node. Oh my god. Okay, and once it's go there, let's say it's go there. Okay, and basically it will present it to the T cell and it will make the immune response. Now the primary infection, as you can see here, this is the CD4. It will be normal, but with time it will drop. Why? See, there is a drop and there is a transient and then progressive loss. Now the drop here is because of the infection and the killing via the virus. That's why it will drop. But your body will recover by producing a new CD4, and then it will never produce, and then it will go down. Now the plasma viremia, as you can see in here. Now why we have too much virus? See, when you are going inside, the dendritic cell will give it to the CD4. CD4, it will be activated, and then it will lead to replication of the virus, because as I mentioned before, virus will go inside. So the virus will go a lot on the lymph node, and then it will be spilled to the blood. And once it's spilled the blood, you will see the viremia. But why is this transient? You know why? It's because of the CD8 and the humoral response of the body. It will try to internalize your virus, and that's why the virus will go down. The CD4, it will not be affected, but why it's going a slowly progressive loss? It's because your body can recover initially, can produce the CD4, but later on it cannot. That's why it go down and then it go to AIDS. Okay. This is the clinical latency. This is basically, I know viremia is very low, I know this, but remember your virus is replicating inside your lymph node. Okay, that's why it's the CD4 is going down. When the CD4 is going down, this gives you indication that there is a lot of virus, but they are not spilling to the blood, they are found in the lymph node. Anyway, now once it go in the critical point, it will go and then viremia will start again. So this is the immunopathogenesis, CD4 depletion. So why there is a depletion? Now we want to understand why. Okay, why this happened. Now, first of all, you can see we will start from here. I put it in the red because I want to start it. Destruction of the CD4. Now they are basically, the virus will affect the T cell. First of all, via this glycoprotein envelope, it will lead to apoptosis. Okay? And we said that the RNA will go inside the infected cell and that will lead to cytopathic effects. Okay? and then it will lead to lysis. The reason why lysis, because once it is infecting it, it will be budding of the virus to go inside the blood. That's why it will lead to lysis of the T cell, okay, or the macrophage. And then at the same time, remember that a lot of surface protein will be found in the surface of the infected cell. The CD8 will kill the cell which presents the virus. That's why it will lead to destruction of the CD4 since it is the infected cell. Lastly, there is another way. It is impaired production. I told you initially there will be sustained production of the CD4, but later on, because of the bone marrow suppression, because of the inflammatory cytokines, remember there is a inflammation in your body, and because of the thymic dysfunction, you will have impaired production, which is basically can be found in here, okay, in the clinical latency. Now, the second thing is the effect of the CD4 dysregulation. Remember that CD4 in lecture number 2, we mentioned that CD4 has effect on the B cell, natural killer cell, macrophage, and cytotoxic T lymphocyte. Now, if the CD4 is dysfunction because it's coming down, okay, the blood CD4 is going down, and because of the infection via the virus, the CD4 will become crazy. Okay, I having integrated viral DNA inside my body. So the CD4, what will happen is that it will not help the B cell. It will not help the cytotoxic T lymphocytes. And eventually there will be no cytokines produced via the CD4 that will help in killing of the virus. Now, the lack of the B cell will lead to the polyclonal hypergammaglobulinemia. It means that the B cell will proliferate, but there is no class switching, and these antibodies, they are not specific to the antigen, okay? And basically the cytotoxic T lymphocytes here, the reason why it will be dysfunctioning cytotoxic T lymphocyte and the viremia will go up is one of the causes that CD4 will not help. It's because CD4 is dysfunctional, okay? Now, lastly is the effect on the CD8s. Now, CD8, how it will affect it? Now, CD8s, early in the disease, it basically decreases, okay? It will lead to decrease of the viral load, but there will be a massive turnover. That means the CD8 will live and die just in order to kill the virus. But the virus will acquire the mutation, and at the same time there will be exhaustion of the CD8, and there will be loss of the help of the CD4, and thus will lead to what? It will lead to the secondary viremia. Now remember, the antibodies that are produced, they are not poorly response to new antigen, so they are not neutralizing, so they have very minimum effect on decreasing of the viremia. Lastly is the one I mentioned to you, how the infections occur. This is how I affect it, telling you. But remember, the follicular cell, which is found in here, once it's still, once the dendritic cell, once it take the virus, it will go to the lymph node presented, and the follicular dendritic cell is a dendritic cell which is found in the follicles of the lymph node in the germline center of the lymph node. Okay. And if it is infected, it can serve as a reservoir, and then the virus will be dormant. And this is all about it. If you have any question, feel free to ask, and thank you.